Hello everyone, and thank you for attending today's webinar, 5 ways to control your critical antibody reagents and avoid bioanalytical assay failure. I'm Tristan Free, Digital Editor of Biotechniques, and I'll be your host for today's event. Before I introduce you to our speaker, I'd like to quickly cover a few housekeeping items. Firstly, all the widgets are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right-hand corner. Please let us know your thoughts on today's webinar by tweeting us at Bioanalysis Zone, or simply share today's webinar with your friends and colleagues using the Share This widget on your screen. Some networks cause slides to advance more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help widget at the bottom of your screen. An on-demand version of today's webinar will be available approximately 1 hour after the webinar and can be accessed using the same audience link that was sent to you earlier. So, we're delighted to have presenting for us today Paul Royal, Technical Sales Manager for Bio-Rad Laboratories Custom Antibody Service. Paul has been with Bio-Rad's Antibody division for over 7 years. Prior to this, he worked in an immunology diagnostics company for almost 7 years. Paul holds a degree and a PhD from the University of Nottingham and has postdoctoral research experience from the University of Warwick. We invite the audience to submit any questions that you may have for Paul throughout the webinar using the Q&A widget to the right-hand side of your screen. We'll pose your questions to Paul in a live Q&A session after the presentation and then follow up any outstanding questions offline. There is also a short optional survey that you can complete throughout the webinar at the end of the Q&A section. This can be accessed using the survey widget at the bottom of your screen. Thank you again for joining us today, everyone. I'll now hand over to Paul to start today's presentation.
Hello, my name is Paul Royal and I manage the Technical Sales Specialist for Bio-Rad's Custom Antibody Service in Europe. And today I will be speaking about 5 ways to control your critical antibody reagents and avoid bioanalytical assay failure. As an overview, I will look at what critical reagents are, and then look at some key areas. Firstly, around suitability of the antibody itself in terms of performance and how considerations such as specificity, format and function can be built into the scope of the original antibody generation. And secondly, considerations around ensuring the ongoing availability of the antibody material such as lot-to-lot reproducibility and stability. A good place to start is to ask what are critical reagents in the context of bioanalysis. A 2018 white paper by the European Bioanalysis Forum describes critical reagents as reagents that require specific testing of newly introduced lots, where a loss of performance can lead to suboptimal results in routine bioanalysis and assay validation. So when we think of critical antibody reagents, we tend to think of capture and detection reagents for ligand binding assays, as is shown on the left-hand side. But for the avoidance of confusion in this talk, I want to slightly expand the definition to also consider assay reference standards, such as antibodies used as serological control standards for immunogenicity assays, where those critical reagents are custom-made rather than patient-derived. So this is as shown on the right-hand diagram. So what does the regulator expect with regards to critical reagents? According to the FDA's Bioanalytical Method Validation Guidance for Industry from 2018, the sponsor should appropriately characterize and document, e.g. determine the identity, purity and stability, all reference standards in critical reagents, such as antibodies, labeled analytes and matrices, and store them under defined conditions. So within this, I really want to pull out identity, purity and stability and align them with the 5 points covered in this talk. With antibody identity, you can approach identity from a few different angles. In this talk, I will have tied the identity with the first 3 of our 5 points, and these are the design of the specificity, the control of the antibody format and the degree of labeling for conjugated formats, and the characterization of binding and function in the desired application. So let's start with specificity. The European Medicines Agency defines specificity as the ability to assess unequivocally the analyte in the presence of components which may be expected to be present. And really we can break this down into some more basic questions. So does the antibody bind the target, and if so, does it bind the target in all of its forms? Does the antibody bind anything other than the target? And does anything in the matrix either negatively affect the antibody binding, or does anything in the matrix directly bind the antibody itself? These questions in mind, we can approach the antibody generation with a view to building these considerations in. So what you're looking at here is a schematic representation of a workflow for generating phage display antibodies. And there are two main areas where there's opportunity to build control of specificity into the library screen process. The first is during biopanning or guided selection, which you can see in the diagram down towards the bottom left corner. So biopanning is a process by which phage displaying antigen-specific antibodies are enriched from the overall library, whereas those phage displaying antibodies that are non-specific are depleted or removed by washing. And what you should then do is arrive at a final pool which is highly enriched for antigen-specific antibodies, and these can be isolated and further characterized. The second stage is during the screening, where these isolated individual clones are further characterized, and those that meet the customer's success criteria ultimately become the deliverables of the project. In the next couple of slides, I want to focus on the first area, really how the conditions under which biopanning takes place can be manipulated in order to meet the specificity requirements. So with this slide, I'd like to address the question that was asked a couple of slides ago. Does the antibody bind the target? And if it binds the target, does it bind the target in all of its forms? So let's imagine a scenario where the analyte is a biotherapeutic, and that biotherapeutic might be present in the sample both in the free form and in a target bound form. And it's desirable to generate antibodies which recognize both forms for total pharmacokinetic measurement of the amount of drug in the sample. So if we were approaching a generation project with these kind of criteria, one of the options available to us is to build in desirable cross-reactivity. And we do this by a process called alternating panning. So with the biopanning enrichment process, we have 3 sequential enrichment rounds. So what we're able to do with an alternating approach is sequentially enrich on up to 3 different versions of the antigen. So in this scenario, say antigen 1 would be the free form of the biotherapeutic, and antigen 2 would be a complex of the biotherapeutic in complex with its target. So as you can see here, in round 1 and 3 we might enrich on the free form, and in round 2 we might enrich on the complex form. And really what this would do is this would drive the selectivity towards those epitopes which are present both on the free form and on the complex form, or certainly that are available to the library on the complex form, because in the complex certain epitopes might be obscured. So this is one approach that we routinely use to generate desirable cross specificity in our antibodies. To address the potential challenges of the antibodies recognizing proteins other than the target analyte, we can actually employ strategies to actively remove unwanted cross-reactivity. So let's imagine a scenario where we want to measure a therapeutic drug antibody, and the antibodies that we're using mustn't cross-react with any other immunoglobulin that might be in, say, a serum sample. In this scenario, we're able to employ something we call the blocking strategy. So here the analyte, or the target protein, is immobilized, as shown in blue, and closely related antigens are used for blocking. So these might be antigens that have a very high sequence homology with the target protein. And what happens is we use these in a higher molar concentration in the solution phase. And so because there's a higher concentration of them, any phage displaying antibodies that are cross-reactive should bind to these closely related antigens, as shown in white. And then because the closely related antigens are not immobilized, they're lost in wash steps. And when employed across all 3 panning rounds, the overall effect of this is to deplete the antibody library of any cross-reactive antibodies, and really only enrich for those phage displaying antibodies that recognize features that are uniquely present on the target analyte, but are not present on the other proteins and the closely related antigens. In the last two slides, we've looked at measures that we can employ to ensure that the antibodies we generate bind to everything that they should bind to, and that they don't bind to anything that they shouldn't bind to. But these aren't the only considerations you may wish to take into account when considering how to design a custom antibody generation project. You may also wish to consider the matrix that your analyte will be present in. So there can be components of the matrix itself which can potentially disrupt non-covalent interactions by which antibodies bind their antigens. So take, for example, urine as a matrix. The normal pH range of urine is 4.5 to 8, and urine contains both salts and urea. Now, since all antibodies are different, not all antibodies will be affected by the matrix to the same extent. And incorporating the matrix, for example, by spiking during biopanning, should tip the selection towards phage-displaying antibodies that are less negatively affected by that matrix. Another consideration is that there may be proteins present in the matrix in which your analyte is found, which may themselves bind the antibodies. So this is really the opposite of your antibody binding a protein it shouldn't in the matrix. This is something in the matrix binding your antibody. So I'd like to unpack this a bit because there are a number of proteins that can have this kind of effect. The first example is HAMAs, or human anti-mouse antibodies. So some human serum samples contain antibodies that will bind mice IgG, and these can either short-circuit your assay or they can mask your capture antibodies, assuming that your assay uses mouse capture and detection antibodies as its critical reagents. Now, at the assay level, you can use something called hammer blockers, which is essentially mouse IgG, non-specific mouse IgG that you spike into your reaction buffer. And this should mop up any hammers and stop them interfering with the components of your kit. I should mention that I'm also aware of an opposite scenario where rats actually have endogenous levels, or some rats actually have endogenous levels of antibodies that specifically recognize human IgG prior to actually being used in any kind of clinical trial. And this can be significant if you're using those rats for early PK or tox work, where your drug is a human IgG biotherapeutic, because those rats will essentially have anti-drug antibodies in their system before you've even administered the drug. Rheumatoid factors are essentially autoantibodies. They're usually IgM class antibodies, and they recognize antigenic determinants on the Fc region of a patient's own IgG usually. And these may also be species cross-reactive, so these can also potentially interfere with your assays. And also, if you're using cells or cell-based assays, many cell types feature Fc receptors on their surface. And this is sort of widely known, and consequently there's a need to block cells or cell-based assays such as flow cytometry, and there are dedicated reagents for this called Fc receptor blockers. Now, in all of the above cases, the issues arise from proteins in the matrix that essentially recognize usually the Fc domain of human IgG Fc. So one way of avoiding these kind of effects and having to use things like hammer blockers or Fc receptor blockers is to actually use a format of antibody that doesn't even feature Fc. So some examples are given below. These are recombinant Fab formats, and also antibodies generated by traditional methods can have the Fcs cleaved off by papain or pepsin cleavage. Okay, that last slide should set the scene for us to seamlessly segue from our first critical reagent focus point, which is specificity, to our second focus point, which is control of format, because eliminating the possibility of matrix interactions is not the only consideration when planning and selecting the format of the antibody that will become your critical reagent. Choice of format is important. So, for example, affinity measurement usually requires a monovalent antibody format to get a nice clean affinity measurement, whereas bivalent formats have a higher overall avidity or functional binding strength. Detection antibodies are usually labeled, and serology controls such as anti-drug antibody positive controls should ideally resemble the native antibodies which will be the analyte in the final assay. And you may want primary antibodies to feature specific species FC domains for panel building to fit in with already optimized species-specific detection methods. So there's a lot of reasons to consider the format. Now, one of the benefits of working with recombinant antibodies is that the antibody format can usually be changed either by cloning, or one innovation that we've recently introduced is the ability to rapidly change the antibody format at the protein level. And for this, we utilize a technology called SpyTag. Now, very briefly, SpyTag is a 13 amino acid tag which is displayed upon the C term of a protein. And when combined with another protein domain called a spy catcher, this will spontaneously form an irreversible covalent isopeptide bond. So we can use this as essentially a tool to very rapidly link monovalent Fab formats to lots of different domains. And by offering a menu of pre-prepared catcher formats, we offer customers the opportunity to receive the Fab antibodies in a variety of formats, and the flexibility to change between formats without the need for subcloning. Now, where antibodies are going to be used as secondary antibodies or detection antibodies, or even, say, capture antibodies are going to be biotinylated for the application, there is a need to consider conjugation. So labeling to enzymes, fluorophores, or carrier proteins is traditionally a semi-random process involving some kind of chemical coupling technique. So most commonly with antibodies, coupling is done to primary amines on the lysines, but there are also other methods such as sulfur hydryl coupling. And such traditional methods come with associated issues. Firstly, these techniques may result in incorporation of the label into the paratope of the antibody. Or in the case of sulfur hydryl coupling, labeling may disrupt the tertiary structure of the antibody. And this may cause issues such as a decrease or even a loss in the ability of the antibody to bind its antigen. And another issue with coupling more generally is that there can be variability in the degree of labeling between different conjugation events, which may lead to lot-to-lot differences, which may in turn affect the performance of the assay. Now, one feature of the SpyTag SpyCatcher technology described in the previous slide is that labeling of the SpyCatcher occurs before the Fab domain is added. The benefits of this are firstly that multiple different Fab antibodies can be coupled to the same pre-prepared catcher, and this should give the customer confidence that the difference they are seeing when comparing different Fabs from the same generation event are essentially down to the performance or the binding characteristics of those particular antibodies rather than the degree of labeling that they're seeing. And the next thing is that because the Fab is added after the label is added to the catcher domain, there will be no labeling of the paratope because the paratope itself is not actually present during the time that the chemical labeling was taking place. And finally, as another step to try and minimize lot-to-lot reproducibility, we've engineered some of our SpyCatcher domains to feature cysteines at specific sites for essentially site-specific labeling using maleimide chemistry. And this is really done with the goal of further improving the reproducibility of labeling. So if we have established the specificity of the antibody and selected the appropriate format or formats, either labeled or unlabeled, another key consideration is whether the antibody actually works in the intended application. So in custom antibody generation projects, a potentially large number of hits must be consolidated down into a manageable number. And this is something that can be built into the screening process so that those clones which look most promising in the intended application can be prioritized. So with an in vitro antibody generation process, incorporating these evaluation steps on different platforms midway through the generation process is relatively straightforward. And the process can literally be frozen if samples need to be sent off to customers for more esoteric testing, for example, in cell-based assays, before a decision about which clones to progress is finally made. And for sandwich pair selection, it can often be quite pronounced how antibodies that all work well by say, direct ELISA may vary in their suitability as capture antibodies. Some simply don't seem to like being immobilized, whereas those same clones work very well as detection antibodies. Therefore, when we're carrying out a sandwich pair screen for customers, we often look at 100 sandwich pair combinations at a time in order to find the best combinations. Okay, so this brings us to the end of the first 3 ways of controlling our critical antibody reagents that fall under the umbrella of identity. So by building specificity, selecting the appropriate formats, and making sure that they're most likely to work in the intended application. And this is all built into the antibody generation process. So now let's look at the next big area of consideration, which is the supply of consistent material going forward. And this also ties into the FDA guideline that I mentioned near the start of the presentation, which names after identity purity as a parameter which should be characterized and documented. So as stated in the European Bioanalysis Forum 2019 publication on practical management of critical reagents, the reagents used for ligand binding assays are often produced using biological systems and may be prone to production variability. Okay, so firstly, let's look at the antibody production methods themselves. Now, just to be clear, I'm talking about production or reproduction methods rather than the initial generation, although for polyclonal, to some extent, the two can be inseparable. Now, with polyclonal, it's important to understand or consider the grade of the antisera and the amount of polishing that goes into it. And it's also important to understand the definitions that some suppliers may give you. For example, what does affinity purified mean? Does it mean that protein A or protein G have been used to isolate the IgG fraction from your antisera, and you, as a result, have a mixture of both specific and non-specific antibodies in that preparation? Or does it mean that the immunogen used on the animal was also used to make an affinity column? The antisera was put against that column, the specific antibodies bound, the non-specific binders are washed away, and then the specific antibodies were recovered, so that all of the antibodies you have in that preparation are specific to your target. Another consideration is that bleeds from different animals may have different epitope patterns, and this may not always be obvious to your supplier. So what do I mean by this? Why might it not be obvious to the supplier? Let's consider 2 bleeds of a polyclonal antibody, which is to say a species-specific anti-human IgG-Fc. So the immunogen will probably be human IgG-Fc from serum, and they'll use this because it'll contain all 4 subclasses, so the resultant polyclonal antisera they raise will recognize all 4 human IgG subclasses. And when the supplier does the QC testing on this material, they will probably use the same Fc material that they used as immunogen, and they'll probably adjust the antibody titer so that lot to lot it gives a similar reading. However, the customer won't be using this to detect probably human IgG from serum. They may be using it to detect, for example, a biotherapeutic drug which is a human IgG4. And when they're using different lots of that antisera, they may see very different performance lot to lot because the subpopulation of antibodies in each bleed, which specifically recognizes IgG4, may be different. For hybridomas, you usually get good sort of lot-to-lot reproducibility from cell lines, although you may see decreasing yield over time. That tends to be the most common problem, and you may need to reclone to find a subpopulation of cells which are higher yielding. With recombinant antibodies, such as those used in phage display, there are a number of advantages, one of which is the sequence of the antibody is defined and usually stored digitally. So even in a worst-case scenario, the gene could be resynthesized and antibody production could begin again quite rapidly. The antibodies themselves are produced by transfection, so plasmid transfections of E. coli or mammalian cells, depending on the expression system you're using. Or in other cases, you can actually make stable cell lines to secrete these antibodies. So really, recombinant antibodies, you have a high level of insight into what they actually are, and you have a lot of confidence in sustainability and long-term availability of supply. So from the reliability of the production method to the purity of the antibody itself, antibody suppliers will, or at least they should, assess the purity of their products, perhaps with the exception of serum-grade polyclonal antibodies. And if you have a requirement for the purity of individual lots to be recorded, this is something that the supplier can often offer as part of a package if you require a more comprehensive set of QC parameters than the standard certificate of analysis or equivalent supporting documentation. So most suppliers will have a basic set of QC parameters that will be their default offering for most projects. And these will be the same for sort of university projects, so they're often at quite a comprehensive but quite a basic level. But your suppliers will probably also be used to dealing with biopharma companies and IVD manufacturers for whom the antibodies that they supply them are critical reagents or critical raw materials in the case of IVD manufacturing. So they probably will be able to supply a higher level of supporting documentation and QC if you require it. So in this slide, you can see an example of Sds Page images from some of our monovalent fabs. So yeah, nice and clean. And if you're wondering why the fab heavy chains are looking a bit heavy, around 30 kilodaltons, it's because our fab heavy chains contain purification tags on the C-term, which are used obviously for purification and downstream applications. Other methods of assessing purity include capillary electrophoresis. So here's one that we've done showing our mammalian expressed IgG formats, and what you can actually see here, and certainly as shown in the right-hand side, is the trace of various possible antibody fragments that are visible under non-reducing conditions. And finally, size exclusion chromatography. So this time looking again at IgG, at the uniformity of 3 lots of mammalian expressed IgG this time. And what you see is nice close retention times, which is what you would hope for. Now, if you have QC or production requirements that are different to the standard offering of the supplier, these are things you should be able to discuss with them and capture maybe in a supply agreement. And this can also be an opportunity to put in place change control notification on critical reagents to make sure that you are notified of any changes or planned changes in process that might potentially affect your product. So now we have covered specificity, format, the suitability in the application, the production method, and the purity of the critical raw material. So this takes us to our final consideration, which is stability. The assessment of stability in the final application is probably the ultimate test that you will need to carry out, particularly if you're making and storing large batches of, for example, antibody-coated plates. However, there are extra tests on the critical antibody reagent that you may wish to order from your service provider. Now, whilst antibodies themselves are inherently stable when either frozen or in the presence of microbial inhibitors, in the sense that the molecules do not fall apart, loss of function over time can be seen with some clones, particularly when we're talking about monoclonal antibodies. And this can be, or is probably most likely the result of aggregation. So assessment of the stability of the antibodies early on in your validation process can help weed out any clones that may have stability issues, and this in turn may save time and energy further down the line. One early indication of the stability of protein is to look at the melting point. So we use a nano-DSF for this to provide data on both the temperature at which unfolding or melting begins and the point or the temperature at which 50% of the protein is unfolded. Broadly speaking, the higher the melting temperature, the more stable the protein. One mainstream method of assessing stability is to look at accelerated stability. So obviously you don't want to wait 2 years before you start using your antibody to see if it has 2 years real-time stability. What you'd want to do is use an accelerated stability study to give you confidence and then run your real-time stability in parallel with other studies. So whilst using incubation across different temperature ranges isn't 100% predictive of the real-time stability, it can be useful for identifying unstable clones. We use the Q10 method to interpret the data that we get from accelerated stability, and this basically states that for every 10-degree increase in temperature, the rate of the reaction, and in this case the reaction is deterioration of the antibody, will double. So using this method, a shelf life for your critical reagent can be estimated. Similarly, whilst repeated freeze-thaw cycles should be avoided by basically planning well and aliquoting into appropriate vials and good storage, deliberately subjecting antibodies to multiple freeze-thaw cycles is another indicator of stability and may help identify if any clones have issues with, for example, cryoprecipitation. Now, I should mention that in this slide and the previous one, both of these studies were looking at the evaluation of mammalian-expressed IgG hucal antibody, and the assay was actually formulated as an anti-drug antibody with the IgG as the positive control. So not only can your critical supplier carry out the stability test for you, they can also do it in a format that may be relevant to your intended application. Some other best practice considerations: obviously maintain an inventory system. So think about how you will both plan and maintain your inventory, and also think about how your supplier might be part of this, since they can hold stock or maybe dispatch material from the same lot to different sites if you have a multi-center study or are working in parallel with a CRO. And also think about the storage of the data, not just the certificates of analysis that come with the physical material that you receive, but also any data from extended QC studies or project reports that you have commissioned. So yeah, these can really help you keep track of all the molecular binding and functional characteristics, all the degrees of labeling, and this is a useful resource because it will allow for quick troubleshooting and sets a bar for reproducibility. Another good practice tip is to store your material in sensible aliquots and in the appropriate buffers. And with aliquoting, certainly when we're providing, reorders of material, we tend to aliquot and then we freeze and then we thaw one vial and we conduct a QC test on that vial, so that we're confident that all the material that we've shipped should be well represented by the lot we've actually evaluated, because it's been through the same number of freeze-thaw cycles as the material will have been when the customer comes to thaw and use that material. And yes, have a plan in place for long-term stability testing. Remember, it isn't just antibodies that can degrade over time with repeated freeze-thaw cycles. Care and planning should also be taken with the antigen that's going to be used in such tests. So I've mentioned engaging with your critical reagent supplier in this presentation before, but I really want to highlight the benefits of this as I begin to wrap up. So you can engage with them to consider the whole scope of the project when you're actually having that initial discussion, when you're planning that initial custom antibody generation project. You can think about the level of quality control you want to carry out on the antibody so that you're sort of most confident it's going to be suitable for your application. And really let the supplier know what reagent attributes are most important to you. You may assume that they can't do anything to influence it, but it may be that they certainly can. And yes, develop a managed solution once the generation's done and you're reordering the antibodies. Obviously, you want to minimize the number of different lots so that you have to minimize the sort of number of lot-to-lot sort of QC or validation tests that you have to do. But equally, your supplier is probably quite happy to minimize the number of lots so that they can have fewer production events, because there's obviously economy of scale working like that. And one of the ways we work together with customers, certainly in the IVD and some biopharm accounts, is that they will order a single large production event with us, and then they will call off from that lot as and when they need the material, so that it's all the same original generation lot that they're receiving, so that they don't have to do that lot-to-lot testing. And equally, we're quite happy with that because we've made one event and then it frees up the resource for other work. So this brings me to the end of my presentation. So just as a way of wrapping up, I just want to touch on sort of what I've covered. So really, the importance of considering the whole scope of the project when you sort of first engage with your supplier, from designing the specificity of the antibody, building into that the most beneficial format or formats that you're going to need to take forward, and the sort of characterization that you may wish to put onto your supplier to give you maximum confidence that the clones that you're eventually sent to evaluate are most likely to work in your application. And then also working with your supplier with regards to continuity of supply, lot-to-lot reproducibility, all the steps that you can make to maximize the chance that the material you get is going to be consistent and that you're not going to have supply issues. And really, if you do all this, if you plan it well, if you engage them, and if you get good results, you should hopefully avoid surprises. You should have confidence in the reagents that you're receiving. They should work well in your assay, and yes, it should help you trust the results that you're generating. So with that, thank you very much for your attention, and I'm happy to take any questions.
Thank you for that very insightful presentation. I hope our audience found it of great interest and value. If you have any questions for Paul, then please continue to submit them using the Q&A tool in the right-hand side of your screen. We've received a number of questions so far, so we'll address some of these now, live on air. So the first question to come in, Paul, is do you make customized antibodies in example for drug compounds to be used in PK assays?
Thank you, Tristan. So for this question, if I understand it correctly, the question's about tool antibodies. So we make tool antibodies routinely as capture and detection reagents which specifically recognize the drug compound. And we also make them as positive control reagents or neutralizing antibodies depending on the customer requirement. Licensing restrictions prohibit us from making therapeutic candidates. So we can't make the drug compound itself, but we can make the tool antibodies to act as critical reagents in the pharmacokinetic or ADA assays that measure those drug compounds. So I hope that answers that question.
Okay, and how long? Sorry, my apologies. And how long does the process take in terms of time?
So assuming we have all of the antigens we need, in-house, in our hands, the workflow is typically 8 weeks for the actual library screen process. But obviously, it depends on sort of what additional services are ordered with it as well. There's sort of more to it than that. But from starting material to delivery of an evaluation sample, the process time is 8 weeks, and we start new projects off every 2 to 3 weeks. So we say 8 to 11 weeks if there's some scheduling time. So it really depends on when the customer material arrives.
And have you done any comparison of the antibody stability between recombinant antibodies and naturally produced antibodies, such as hybridoma, especially for rearranged different isotopes?
I'm not sure if we have any sort of direct side-by-side studies where we compare two together under exactly the same conditions and assay them together. But we do have, if you look on our website, there's something called critical reagent. We have a flyer about critical reagents, and that shows some stability studies we did on IgG formats and Fab formats. But I think this probably ties into another question as well. With our standard Fab products or our IgGs, what we say is they should have at least a 2-year shelf life when stored at -80. But obviously, we make custom antibodies. So for a custom antibody, unless the customer sort of specifically asks us to do it as an extended QC service, we're not going to sort of do a real-time or an accelerated stability on sort of antibodies we've literally just generated as a custom deliverable for a customer. But what we know from our catalog products and our sort of past experience of reagents that are reordered for IVD manufacturers, they're sort of as stable as normal antibodies as far as we're concerned.
And have you compared the performance of SpyTag antibodies with standard antibodies?
It really depends. In terms of, it really depends on the application. They can be used as capture antibodies, they can be used as detection antibodies. What I would say is, in terms of how comparable they are, in terms of naked antibodies such as capture antibodies, they're very comparable. In terms of labeled antibodies, we tend to see a higher activity with SpyTag versions because, well, we think the reason is because there's no incorporation of the label into the paratope of the antibody.
And what are your timelines for preparing the critical reagents? Is it 2 months or 6 or 12 months?
As I mentioned before, our process is 8 weeks typically. So this is for a new generation. If you've already done a generation project with us and it's a reorder of raw materials or we're just simply doing a production run, the timeline can be anything from 1 to 2 weeks for a fast track small volume production to sort of 4 to 6 for sort of 50 to 100 megs. It's a very fast process, assuming it's E. coli expressed. We simply transfect and purify. There's obviously the size or the quantity requirement will determine the size of the culture and the timeline, but it's pretty fast to be reordered. And I think I answered that question earlier in terms of the original generation taking 8 weeks, but we say 8 to 11 because we have up to 3 weeks scheduling.
Okay, and in terms of your expiry data, would you set expiry date to 5 years based upon the King paper?
We haven't changed it to 5 years. I'm not sure if we plan to be honest. Essentially, we send our products out preservative-free and frozen in phosphate buffered saline because customers can use them for all sorts of applications. Some of them are, say, in cell-based assays where you don't want preservatives. Or they may wish to conjugate themselves, so they may not want anything like stabilizers or primary amines in there. So we say, at least 2 years unstored at -80, but they're antibodies, they're inherently stable, and it is something we very, very rarely get any sort of queries or complaints about.
Okay, and next up, do you think that the production of capture reagents based on the COF ranking guarantees a good performance of LBA methods?
So could you just repeat that question, the capture reagent?
Yeah, so the question is, do you think that the production of capture reagents based on the COF ranking guarantees a good performance of LBA methods?
Okay, yeah. So it doesn't guarantee a good. No, KOF ranking doesn't necessarily guarantee good performance because not all antibodies work well as capture antibodies. I think I mentioned it in my presentation that some antibodies don't seem to like being immobilized. So and obviously KOF ranking, we immobilize the antigen to do that. So the antibody itself is not immobilized. It's a pretty good way of essentially, or the main reason we use it is to stratify a large number of hits or consolidate a large number of hits down to a smaller number based on their off rate. And usually customers want us to select the clones with the slowest off rate because they're the highest affinity binders. So, if they do work as capture antibodies, they will probably give you a better analytical sensitivity if they're higher affinity. But as I say, it doesn't sort of guarantee their suitability as capture. But I suppose another thing that KOF ranking is useful for is if you're making anti-drug antibody controls. Some naturally occurring anti-drug antibodies are relatively low affinity, so we can actually choose sort of antibodies with slow off rate as the sort of high affinity ones, but we can also take some with sort of medium and fast off rates if you wanted to have, say, anti-drug antibody controls across a different affinity range to see if that's more physiologically relevant to your assay.
Okay, and what kind of affinity would be expected in general for the antibody from phase display?
So with our library, it's pretty good, but what I would say is it depends on the stringency of the library screen. So if it was simply, here's a protein, generate an antibody to any part of this protein, what you'd have is, within the library, you'd have a very large number of antibodies which recognize the different epitopes on the protein, so there'd be a lot of competition and the higher affinity binders would win out. So in that case, we'd if it was literally here's a protein, we'd expect single-digit nanomolar affinity, or maybe even sub nanomolar. It's really quite good for our library. if you're literally looking at a single post-translational modification or a mutation or something very small, there'll be a relatively small number of antibodies competing for it, so the lower affinities can sort of come through, and there you might have double or even triple-digit nanomolar affinities.
And what do you think of storing the antibody with BSA or azide-free at 4 degrees centigrade refrigeration? Do you think it is a stable for storage up to 1 year in these conditions?
If you had it azide-free just with bovine serum albumin, because we supply it in phosphate buffered saline, you wouldn't have any preservatives in there, so you may get microbial contamination, that can cause very rapid degradation of your protein if the microbes are releasing proteases. So I wouldn't recommend storing it with BSA azide free. I would put something in there. It may not have to be azide. It could be something like thimerosal, but you'd probably want some kind of preservative. And yeah, at 4 degrees, you can leave antibodies at 4 degrees if they're preserved, and if we don't see a huge amount of deterioration. But the best thing to do really is to get them, aliquot them, freeze them, and thaw them prior to use. That way you'd be more confident of not getting any issues. And just on the subject of BSA, for any companies that are based in the UK, I would just say there can be issues with shipping material that contains BSA into Europe now with Brexit. That's just something we've noticed, that it's only a stabilizing protein to put into your thing, but it can cause, because it's animal derived, it can cause some Brexit shipping issues. So if you have an alternative protein, it might be better putting that in a non-animal derived one. Sorry, I've gone off on a bit of a tangent there.
Okay, well, continuing to look at the sort of temperature of storage, can all antibodies be frozen?
Yes, as far as I'm aware. I mean, there may be some conjugated versions where it may not be advisable to freeze them, but the actual antibodies themselves, yeah, our standard way of making them is we make them, aliquot them, freeze them, and then we dispatch them on dry ice. So that's our standard method of doing the Hucal antibodies, at least our recombinant ones, because, as I mentioned, they're preservative-free and phosphate-buffered saline. But for catalog antibodies that do contain preservatives, we can send them at ambient temperature. So they are very stable, but we recommend freezing them just because of the format we prepare them.
Okay, and are the sequences of your custom antibodies available?
Yes, they can be. So we obviously know them all because we sequence as part of the process. We can sell them to customers, and with that purchase comes permission for the customer to make them themselves in their own expression systems. So I think the UK price is £2,000 for a sequence at the moment. And so, yeah, you can buy that sequence. You can buy the expression plasmid as well from us. And as I say, with that purchase comes permission to make it yourself.
Okay. And one of our listeners writes, great point on BSA and shipment issues. What alternatives would Paul suggest?
We haven't put one in place ourselves yet. I mean, I know there's a company in Nottingham that makes recombinant human serum albumin, so maybe that would be a possibility. That company is called Albumedics. But yeah, I guess it's not something I feel fully qualified to answer at the moment, but anything recombinant or human derived to get around the sort of veterinary import side of things would be my recommendation.
And how do you determine lot-to-lot consistency of your antibody?
So we sort of look at the purity. We look at functional binding. Usually we still have enough antigen left over from the generation. So we, and we give a protein concentration, but it's a fairly basic comparison. Really, what we're checking is the antibody works. We don't routinely compare it to a previous lot unless it's a catalogue product or unless we have an agreement with the customer or they've ordered an extra level of QC. But as I mentioned in my talk, it really depends on the requirement of the customer. So we can prepare a reference lot. We can, usually what will happen is the customer would like us to prepare 3 separate lots because they want to do some kind of validation. We'll take one of those then as a reference lot, we'll aliquot it, and then we can compare all subsequently manufactured lots to that original reference lot. So we'll do a side-by-side, usually a titration ELISA, so that we see how equivalent the two are, and we'll maybe set some parameters for sort of pass-fail based on that.
Okay, well, thank you very much, Paul, for answering all of our audience questions today. Before we wrap up, do you have any last comments that you'd like to add?
Just thank everybody for their attention, and yeah, feel free to get in touch if you have any specific projects you'd like to discuss or any further questions relating to the presentation. Our role is essentially to be the interface between our customers and the laboratory in Munich that does this kind of work, and yeah, we're always happy to sort of discuss needs and requirements and anything else. So, and obviously thank you to you guys for organising it. Oh, we have another question, I think it's just come in, sorry. Can we freeze the antibody with glycerol?
Sorry, yeah, one final question is can we freeze the antibody without adding glycerol?
Yes, yeah, we don't add glycerol when we freeze our antibodies. It's just phosphate buffered saline.
Okay, fantastic. Well, thank you very much, Paul. I'd once again like to thank you as our speaker and presenter, as well as you, our listeners, for your time and questions. If you would like to let us know your thoughts on today's webinar, please feel free to complete our feedback survey in the resource widget or tweet us at Bioanalysis Zone. Don't forget to visit us at www.bioanalysishzone.com for more webinars. Thank you again for attending, and goodbye.